Dear colleagues, welcome to our November dialogue, to our Socratic dialogue. You will enjoy a dialogue with a prominent, famous European cardiologist, a legend of European cardiology. When you will hear his name, you will understand that this is a special dialogue. And this dialogue will be about past, present, and future of troponin, but also biomarkers, but will be also about the human experience of our famous guest. I think that by now you can guess perhaps the name of our famous guest. You know, troponin has many fathers, but only one mother. And the mother of troponin will be with us today for our dialogue. His name is Hugo Katus. Hugo, welcome. And now it's my pleasure to ask Susanna. Susanna is Associate Editor of the European Heart Journal. I would like to ask Susanna to present Hugo Katus, and then we will start our conversation. Please, Susanna.
Thanks, Filippo, and genuinely thanks for everybody who's dialing in and also everybody who's listening after the event. It really genuinely is a privilege to introduce Hugo Katus, who is one of the most influential figures in modern cardiology. So I've learned a lot about Hugo over the last few days. He was educated and trained in Heidelberg, and then he spent some formative years at the Massachusetts General and Harvard Medical School. And what we know about Hugo is, of course, he pioneered cardiac biomarkers and led the development of the troponin T assay, which is a discovery which genuinely has transformed how we detect and manage myocardial infarction worldwide. So in addition to that, Hugo's held chair positions in Lübeck and Heidelberg. He's built multiple centers of excellence across a range of cardiology, so cardiac imaging, cardiomyopathies, cardio-oncology, end-stage heart failure, and rhythm disorders. So that's the simple stuff. But beyond academia and cardiology, Hugo's done a number of things which I think really do matter as we step into this dialogue. So Hugo has served as president of the German Cardiac Society, and he founded the German Cardiac Society Academy for Cardiology. And then amongst many, many ESC activities, he way back chaired the ESC Advocacy Committee, which we're going to return to later. And he currently chairs the ESC think tank on innovation. Hugo's also, so listen up for top tips, generated millions in competitive funding. He initiated the Informatics for Life digital transformation program with related Smart Heart Center. He co-founded 2 gene therapy companies and is still recognized and continues to be recognized amongst the world's highly cited scientists, not just cardiologists, but scientists. And today, Hugo continues at Heidelberg as a senior professor, chairs the scientific committee of the German Heart Foundation. And for me, it's really difficult to imagine modern cardiovascular medicine without Hugo's impact and actually ongoing impact. And it is a genuine privilege to have this opportunity to have this dialogue with him today. So Hugo, really it's a privilege to have you. Very much warm welcome.
Thank you.
So when you finish blushing, let's get down to
it. Kind of you. Thank you very much.
No, much deserved. So I'd just really be interested, Hugo, why cardiology? So your early work, as I understand it, focused on experimental electrophysiology and arrhythmias, but why cardiology of all the things you could have done?
Well, that's a strange question, actually, amongst cardiologists. So I was always excited by the heart. I was, even as a student, impressed the continuous work of this organ and the dependability and this close connections with emotions and adaptation to physical stress and emotional stress. So it was very, for me, fascinating to have such an motor and continuity in strength and rhythm. So that's more about why I went into cardiology. Now with arrhythmia and my thesis, it was more on on anaphylactic reactions in guinea pigs, and we studied the impact of histamine release on arrhythmias. But more importantly, we studied at that time membrane currents and the connection of membrane currents to contraction, so electromechanical coupling, which was in the 1970s a very, very innovative type of science. And actually, I continued with investigations on sarcomere proteins and contractile function when I then really started my clinical work. And there I joined a hemodynamics research group. And since at that time in clinical research, contractility indices and the assessment of contractile reserve of a diseased heart was really a hot topic, and doing pressure volume analyses in patients and stressors to really detect contractile reserve of these hearts was fascinating. So I have stayed in that world of contraction and heart function, and not so much arrhythmias, but and consequently I worked, if you want, so on sarcomere proteins as biomarkers. I worked on sarcomere proteins in cardiomyopathies. I work on sarcomere proteins when we develop also gene therapies, and so it's really a bit focused on contraction and strength development, which fascinated me from the beginning.
And Hugo, then you shifted obviously towards the acute coronary syndromes and the biology around infarction. Obviously, there are links there, but that is quite a major shift. Was it by chance, or was it by design, or was there somebody who said, this is what you should be doing, especially for the younger people who are listening in? What was it that nudged you in that direction?
Yeah, it was not a shift, it was a disruption, actually. I mean, it was by chance, actually. So, you know, I decided for myself when I started my clinical career that the mix of research and clinical care, that's what I'm aiming to. And I found that very fascinating, being on top of science and still being a physician. So, and there was an unwritten rule in our department said who really want to go for an academic career, he must leave clinical education for at least 2 years and go abroad. And that was what I really asked my former boss, Kübler, to support me in finding an appropriate place in the US where I could do my studies. And then, really by chance, I was asked to come upstairs to the boss's office, not knowing what I had to expect. And I still recall that time. And I found in his room then also Professor Arnold Katz. Arnold Katz was a very famous muscle physiologist. He did his sabbatical at the Max Planck Institute in Heidelberg, which had a focus clearly on calcium and calcium electromechanical coupling. Then, so they were thinking, what should they do with that young fellow who wants to go to the US? And I proposed he is going to suggest some muscle physiologists in the US, but he suggested Edgar Haber. And he argued that Edgar Haber, in his opinion, is the most innovative scientist since, and he quoted, Edgar was the first physician to apply antibodies for treatment. And he did that to treat digitalis intoxication. And so that was the first application of antibodies for care long before rheumatology and oncology and all these disciplines are using these antibodies. So Edgar was a pioneer on that. And so I applied in his research group, and he luckily positively responded and proposed that I should work with Banan Kaur's group in his lab, who focused on anti-myosin antibody imaging. So the binding of antibodies to necrotizing cardiomyocytes in the sarcomere and using labels with technetium or different labeling methods. And so I applied for that, wrote a grant application to the German Research Foundation, got funding, and then took the family and stood at the Logan Airport. No hotel reservation, no dimes to call for any hotels. And so we were totally naive and with a child. And so the beginning was very, and I would never do something like that again. But that's how we did it. So we just started.
So I'd say, thank goodness you did, Hugo,
Yeah.
would be the first thing I'll say.
Yeah.
And you know, thanks to your long-suffering family, no doubt, for having put up with probably a very difficult disruption at that time. So apart from obviously the people in your home and your family that have surrounded you, you've worked in some really different healthcare systems and really different areas. And it sounds like there's a bit about opportunity and there's a bit about specific people. So if you were to look back and say, you know, think about your time in Heidelberg and think about your time in Harvard, who would you say were the most influential people who, and then maybe beyond, who've really shaped your scientific trajectory? Who are the people that have really got you thinking, fired you up, and kept you at the top of your game for so long?
Yeah, if I may just start with the first point you mentioned, I mean, that change of my academic education for muscle physiology and contractile research and hemodynamics to antibodies and imaging. I never had used an antibody. I never knew how to purify antibodies. I never purified any proteins. I haven't done any infarct imaging before. So, but I really would, since I'm convinced it was a good idea, since I was able to bridge my knowledge in muscle function with the biochemistry to muscle and clinical chemistry to muscle. So my point is, change, at least in education, your methodologies once in a while to bridge different sciences or approaches to science. And that's what I did. Was not so easy, but it worked at the end. Now, always you depend on mentors and people who support you. And I really enjoyed Professor Kuhn. He was my mentor in my thesis. Very human physician, very patient-centered specialist in muscle disease and cardiomyopathies. So he was, for me, a role model of the best physician to have. So you could trust on this person. And then I got that position in Wolfgang Kübler's group at the University Hospital, medical center. And he was very different. He was a very leading by example, very much disciplined, very hardworking, and very strong and sharp in his thinking and also in implementing structure. So he was a little bit opposite. He was a manager type more and a strong person, but again, a very fascinating person. And his saying always was, nothing said is also praise. So let's just so this to show his thinking. And then it was Edgar Haber. Edgar Haber was, you know, he was a basic scientist, but he was chief of cardiology. He saw one patient or two patients per week. And he worked on protein folding, and his chief at NIH at that time got the Nobel Prize for his work. And he worked on antibody diversity as a cardiologist. What makes a binding of antibody to an epitope? And he discovered renin together with Victor Sau, who was a colleague of him in his lab, and a colleague to mine also. And so he was a quiet listener, thoughtful, and still ambitious and straightforward. So very different personalities, which all, in a sense, were complementary for me, and I learned from every one of those. Yeah.
So, Hugo, I now have to ask you, and then just before I pass over to Filippo, so excuse me for one more question, Filippo. I'm going to just ask you about troponin. So fundamental. Everybody knows about it, everybody learns about it. But at what point in your troponin journey did you realize the real potential that this might be such a game changer? Was there one moment where it dawned on you that actually this was a big deal, or was it something that came over a long, long time?
Well, we had a clear hypothesis, and I think you really need to know where you want to go, and you need to know what your aims are. And our hypothesis, and that was from infarct imaging with myosin, I see seeing so much sarcomere sitting there, knowing from my muscle physiology that there are isoforms, which may be identified by different antibody molecules. So our hypothesis was clear that if antibodies go inside and bind even after day 7, since there's still myosin sitting there, then there will also be release of myosin even on day 7, right? Or fragments of sarcomere. So the hypothesis was there, and and luckily at that time, there was an invention of monoclonal antibodies by Cesar Milstein and George Köhler, and our lab, or Edgar Haber's lab, was very closely connected. So we were one of the first to apply this technology. And the advantage of that technology is you just hybridize one cell, antibody secreting cell, with a plasma cell. So you immortalize an antibody producing cell forever. And you have one antibody for one cell. So you can now have 10,000 cells and 10,000 different antibodies. And that gives you the potential to engineer your assay, okay? And we invented at that time the basic foundation of double sandwich assays. So that you bind one antibody molecule to test tube or solid phase, and that antibody is selected to affinity and specificity. And this antibody will catch your myosin light chain or troponin or whatever you want to measure and sticks to the solid phase. And then you use another pre-selected, clearly defined antibody and label that with radioactivity or enzymes and detect in such a way the antigen. And this is a basic principle until today, how assays are manufactured. We had to Harvard applied for a patent on that, what we developed. Long court decision, Genentech won, unfortunately, at that time. But that did not interfere with our scientific. But it was important that we then also had the tools to measure highly sensitively protein mass. And that was the second point where we were convinced that we will be better than enzyme activity measurements by CK or CKMB or whatever was the gold standard at that time. So we were convinced. And then a more important part was that we joined, actually we were forced to join with industry. Since when I returned back to Heidelberg, still in clinical training, I was a young cardiac fellow. So, and we aimed to develop that new assay. And so we joined with Boehringer Mannheim. They were a bit reluctant since they were the global leaders in clinical chemistry testing for myocardial infarctions, the CKMB stuff. But the advantage having them with us was that they are global leaders in infarct diagnostics and have their experienced team to constantly improve our assays. And that was, again, support where we felt we are going to manage that. And then importantly, we had worked with myosin light chains and developed myosin light chains, patented that also successfully. And Boehringer had developed an assay for myosin light chain jointly with us. Packages were ready to be sent off, and we had realized already half a year before that troponin is more specific than the cardio-specific myosin light chain. And then I had to go to Boehringer and say, we should not sell myosin light. We need to start from scratch. And that was, so they stopped cooperating with us. But we continued and then on troponin and selected troponin T. And at that time, also unfortunate for us, one of your colleagues in UK, Cummins in Glasgow, muscle physiologist, had used a very crude, I say crude assay for troponin I, and tested troponin I following our work with myosin light chains in a canine model. And so we could not get intellectual property on troponin I, but only on T. So that's the reason why it's only one T, but many, many I's on the market. Yeah, but we then could convince again Boehringer Mannheim that they really need to invest in troponin T, and then we were convinced it will work. But I tell you, what we really underestimated, totally underestimated, is the resistance to novel procedures in a community. No one was interested in our work. No one. It was. Our papers were rejected since we were confusing the community. Everyone knows how to diagnose myocardial infarction, and the translation of a novel idea in clinical practice is not so easy. It's not so easy, and we were again lucky to have a strong partner on our side, Boehringer Mannheim, and within Boehringer Mannheim there have been people from therapeutics. Boehringer Mannheim was also doing thrombolytic trials at that time, and beta blockade, and so they came, and we were the first biochemistry group to go to randomized control trials to evaluate troponin. And so we went to Uppsala, we went to the Gastro guys, and also to TIMI people, and they could confirm independently of us that troponin release in unstable angina is actually a surrogate of microinfarction and unstable plaque, and is to be diagnosed as STEMI. And it took us 20 years to get into the guidelines. 20 years to get into the guidelines, and many objections. And these objections never stopped. Since troponin, when you look at the chest pain setting, most troponins are not due to an acute coronary syndrome. So we really had to learn how to deal with that, and had to convince, and that will be also a change. I mean, we had been thinking of developing a biomarker for myocardial infarction, but in fact, what we are developing is a biomarker for myocardial injury, right, or for myocardial remodeling. And it is a clinical setting which defines, is that biomarker due to myocardial infarction? Is this biomarker due to myocarditis? Or is it biomarker due to hypertension remodeling? And so people have to learn that we are not providing a diagnosis for myocardial infarction, but an indicator of myocardial injury, even of senescence of myocytes, which also is a dying process. And so people have to understand that it's a different mechanism. And people also have to understand not to dichotomize in AMI and not AMI, since it's a continuous, it's on scale, has an impact of troponin. So these complete changing in clinical perception of how we approach infarct diagnostics and assessment of myocardial injury, that was not so easy. That wasn't. troponinitis or troponin bumps, and all these terms were all very common to us. Yeah, so it was not so easy. The development was challenging since I didn't know how to purify the protein. We had so many problems in test development, but the clinical translation was equally difficult and actually also emotionally not so easy, since you provide your assay, which you didn't really evaluate yourself, to big groups, really, to Gusto and Uppsala, and say publish then in New England Journal. And I have been rejected with my development and had no option to be published in circulation, right? And you are not quoted anymore. So, but you know, that's part of the business, right? It's part. If you want to have success, you need to share, and people need to love to work on that project. Otherwise, you won't distribute. So it's yeah, it was sometimes challenging, but at the end it worked, and that's important. But we were always convinced that it's a better tool than CK, and you know, and there was many people with CK, and they defended the CK. Alan Chaffee, you know, American guys, and so it was not so easy. It was not so easy, and again, it had only a few people who really loved novel things, like Christian Thyssen, for example. So the Nordic countries loved biomarkers also at that time. So they were more enthusiastic, and Christian, together then with Joe Albert, started that universal definition group, which helped a lot for us also.
So Hugo, I mean, that's what a lesson in history, persistence, and actually being right is the fundamental thing. But I think also a lesson is that the humility that you have to have at times to be generous enough to collaborate and watch other people taking the glory for something that you've spent decades developing yourself is quite extraordinary. I think a lesson for us all. So thank you very much for sharing that with us. I'm now going to pass to Filippo for a bit, who's going to delve, I think, a little bit deeper into troponins. Filippo.
Well, thank you, Susanna. I really enjoyed listening to your academic life. And you know, we had more than 40 dialogues with top cardiologists from the 5 continents. And I can identify a pattern by now. The choice of an academic career is mainly driven by passion and curiosity. And really, you have expressed this notion very, very clearly. But then when we come to the topic A, B, or C, is more by chance than by design. It happens something in your academic life with your mentor, and you meet unexpectedly somebody, you switch on something in your brain, and then that becomes your life. So the choice is passion and curiosity, but the specific topic is more by chance than by design. And this theme recurs in the life of many top cardiologists in the world. Now, coming to the question, academic life is difficult. I mean, again, passion and curiosity, but we have difficult moments. Can you share with us the most difficult moment in your academic life? I think that this can be very useful for our younger colleagues, and also in general. What is the North Star in your life? What inspires your life in depth?
It's very yeah. Well, and there were many. I think if one may start this way, you need persistence, as you said, and you must be convinced that you are on the right track, and then you don't care about opinion of many others, right? and said you need to follow and you need to really focus on a topic. If you want to have a breakthrough, it's not by pushing the whole wall, but by just a very minor point that you can achieve a breakthrough. So we had many difficulties in developing our research and bringing it into translation, but I would rather focus a little bit on mechanisms that are very helpful when you have these difficulties. So one very important point for me was that the principle of having success depends very much on a good team, right? Good team in your research, a good team in your clinical practice. And so it was very important for me always, particularly when it was difficult and many challenges, also in the development of the department or stuff like that, that I had a very dependable team on my side. And the leadership of a team really depends that you have a consent with your team. And so we really tried to have a really joint venture always in a team and try to be a leader in consent with the team around. And the second important point for me also was when you are developing new strategies, be it a new test or being a company or being a chief of a cardiology department, you are being an entrepreneur. You are being responsible. And one of my also famous patients, and I can't unfortunately not name his name, he told me, listen, he was a really very successful entrepreneur. He told me 3 tasks of an entrepreneur. First, implement innovations, okay? Second, take responsibility. Take responsibility. And third, lead by example. So, and we try to really live that. So we invented not only troponin, but also when we went to in clinical practice, we said, well, what do we have an advantage of troponin if there is not an environment where it's being applied? So that's why we really then established as first a chest pain unit in Heidelberg, which is now common across Germany. And when we worked on cardiomyopathies, we developed an institute for cardio. So it means that you really must implement innovation and you must take responsibility if things don't work. And it's always the fault of you and not only of your team. And so working hard jointly with the team was very interesting. Then an example, when we were working with troponin, trying to prove that troponin is cardiospecific. Okay, so we had the idea, how are we going to prove that? And then we decided we have an Olympic institute in Heidelberg, and we asked, how can we induce a skeletal muscle injury? Okay, and say it's isometric exercises, right? So all my lab team and all my co-workers were requested to go to this Olympic center and had to do isometric exercises. And then I received the CK measurements of everyone. So my CK was 30,000. And some of my co-workers was 200, okay? So that's one of the examples where we really try to go jointly. And it was interesting that and those who were with 200 were not so very successful in research. And those with into 10,000 were really so hard working. So what I mean to show by this example is just that you need to have a team and you must remain in it and go jointly. And have consent. So that's very important. And then you will master many difficulties with an appropriate attitude. You need to have an attitude as a scientist and physician. You need to have perseverance, not narrow-minded. You need to have courage. You need to have willpower, and so you need to provide to your environment a leading example, and that's I think is very important when you go for such a journey of academic medicine and clinical care.
Thank you, Hugo, for sharing your wisdom with us and with our younger colleagues in particular. During the dialogues, we want to unveil the human side, because when we come to leaders like you who really have changed the history of cardiovascular medicine, I think that the way you reach your results is a source of inspiration for our younger guys. Now, let's come back to our daily clinical practice. As you said, troponins now are well established in guidelines. But what about the future? Do you think there's room for a dual biomarker strategy in patients with suspected ACS, or will it be possible to beat troponin?
Yeah. Well, when we focus on acute coronary syndrome and consider that there is continuous progress in technology of assays and precision, and you will see even more precise and more sensitive troponin assays, it's very hard to beat a troponin test. Then the question is there myocardial injury, yes or no? So I think there is no added place. There was some moments where we had the problem in the early phase of acute myocardial infarction, where we need earlier marker like myoglobin or fatty acid binding protein. But with the more sensitive assays, these markers are of no added value. You may have need, when troponin is entirely normal, of a vasopressin measurement, so copeptin may have some added value in combination. But again, there is an enormous continuous effort to improve troponin. So I'm not so certain. And there is myosin binding protein, which is very similar to our myosin light chains. But again, I do not expect any added benefit. And dual testing makes much more sense for trying a site of detecting myocardial injury, investigating in the causes of the injury. So is it atherosclerosis, myocarditis, is it pulmonary embolism? So that's more but not for detection of ACS. And yeah.
Thank you, Hugo. Very, very clear. What, in addition to biomarkers, now we have artificial intelligence. You think it's more promising than other biomarkers in this setting?
Well, what is the future of diagnostics in ACS? And then you really need to consider the progress also in deep phenotyping of the heart by other methodologies. And AI-assisted ECG is clearly a progress. You have also progress in imaging technology. And then you use AI to really bring all the data together. So high sensitivity assays with an AI-enhanced ECG, and Roche is joining actually partnership with PM Cardio. So this AI-assisted ECG will be joined with troponin T measurements. And then you will have also algorithms that are more integrative, that integrate, since you have with age, your troponin increases. With sex, there are differences. With pre-existing cardiovascular diseases, there are differences. So this background noise must be differentiated from a true novel signal. And there will be a more personalized approach with AI very helpful. So I believe, yes, we will see much more integrative strategies in biomarker testing, not only focusing on biomarkers. For biomarker testing in the future, it may be interesting to think on markers that catch the disease pathways. So we tested, for example, signatures of microRNAs, or now methylation patterns in blood indicating different maladaptive processes. So that may be a topic, but it's not so easy to standardize these omics and signature and pattern analysis. So what is also, when we look at the future, must be considered, we will have more and more identification of the causes of diseases. I mean, atherosclerosis, you have said inflammatory pathway. So we may have not only CRP, but markers that indicate specific inflammasome pathways to be targeted by therapeutics. So, or if you have a mutation, then you have so these companion diagnostics will increase with more possibilities in targeted therapies. Still, there remains a problem with innovative biomarkers, and we were lucky with Boehringer Mannheim on our side. It's for a startup company, it's very hard to fulfill all regulatory studies, and particularly for biomarker innovation, that's not so easy. So I'm a bit. There will be slow progress. I think the fastest progress will be in joining data and use AI for analysis.
Thank you. Let's come back to your baby. Is there life for troponin beyond the emergency department?
I'm sorry. Yeah.
Is there life for troponin beyond the emergency department, in other directions, in other settings?
Yeah, that was one of our also main challenges. It is, as I said, a marker of myocardial injury, and when you have an acute condition, be it myocarditis or be it sepsis or be it any bleeding or so, or do you have chronic conditions with hypertrophy or any different cause of myocardial injury, then you will measure elevated troponin. And we know that when troponin is elevated after general surgery, it has prognostic impact. And not only in acute conditions, but any elevation of a troponin, also in a non-ischemic setting, has impact for outcome. And it will trigger some cardiovascular examination, not invasive examinations, but cardiovascular investigation. So it's a very sensitive indicator of involvement of heart in any disease process, cardio-oncology, checkpoint inhibitors, and so on. So that it will become standard of care to screen troponins in other critical conditions and in other chronic conditions and in inflammatory diseases and so forth. And it's also interesting that since it's a marker of remodeling, also that when you treat a heart failure patient, and treat him properly, his elevated troponin will decrease. So you have that change. It's not a persistent elevation. So you can monitor your disease and also the impact of therapies. And when you look at presumably healthy population, you have these patients who have elevated troponin, and they have a poor outcome, cardiovascular mortality and total mortality. And when you look at a chronic coronary syndrome patient, 20% have elevated troponin. They have unstable lesions. And I predict we will see trials showing that in chronic coronary syndrome, if troponin is normal, the aspirin therapy may eventually not be beneficial. So we will see these more and more trials on the value of troponins outside of the ACS diagnostic problem.
Thank you, Hugo. Very, very informative, very clear and inspiring again. Well, in the audience, I see a famous Italian pathologist, Eloisa.
Oh, Eloisa.
Eloisa is there.
Hey.
Family reunion.
That's an honor. I mean, we didn't talk on cardiomyopathies. Otherwise, we should have talked on Eloisa. I mean,
okay, Eloisa, you have a question for our famous
great,
guest?
Great. I know, and I call you the troponin man. When I say Hugo is the troponin man to my young workers. Beyond the maya, Hugo, do you think that myocytes that release troponins are irreversibly injured or not? Because my idea is not.
yeah.
I'd like to know your opinion.
Nice question.
Since you are such a wise person, and you are right again. So we have more and more evidence that there is release of troponins in small vesicles, plaques formation, which appear under stress conditions on cardiomyocytes. And when you look at atrial tachycardia, or I think it's the same mechanisms apply. What is very interesting also, what we not studied so well is that we do have different ways or paces in senescence of cells and injured cells, which also in cardiomyopathies is very important.
Yeah.
And so when we measure troponin in these heart failure patients or in elderly patients, eventually we have a good indicator on assessing the biological age of the heart, right? Not the chronological, but the biological age. So there are many fascinating ideas still around troponin. It's not all with ACS and cardiomyopathy and heart failure. They're also very interesting. Yeah.
Fully agree. Thank you. Thank you, Hugo. Nice to.
Thank you, Eloisa. Susanna, back to you now. I'm sure we have additional questions.
Yeah, I have. It's been fascinating to listen. But one thing that strikes me, Hugo, is you're the master of the long game, it seems. And you've been previously, so I'm currently have the privilege of chairing the ESC Advocacy Committee, but you are a previous chair. And you set in motion a number of things that I absolutely genuinely believe that have led to what we now are luckily beginning to see, that for the first time in 30 years, cardiovascular disease, which is the biggest killer across the EU, is going to have a cardiovascular health plan. And I know that was something that you championed when you were chairing the advocacy committee. So looking back on this sort of very strategic thinking and the long-term view, so you don't necessarily see the benefit yourself, but down the line, the benefit will be there. Do you have any lessons from when you were leading that? You've talked a lot about leadership, and any lessons for younger people going forward? I know you're a fantastic mentor to many people yourself, but in the advocacy space, do you have any big lessons, and what do you see as the big challenges facing cardiology going forward?
Yeah, yes, thanks. First, I'm very grateful that you are now leading advocacy, and congratulations for the health plan. So that's very, very, very important. Now, I had been involved in advocacy since I founded that Academy of Cardiology, and I also served as president of the German Cardiac Society. And I was always embarrassed that we in cardiology use the wrong messaging. I mean, we say so many deaths due to heart diseases, but who can care with so many deaths? We need to care with a single person. Only when you look at a single person and understand the problems in the family, when the father has some severe cardiac disease or a child has a severe, then you understand suddenly, and then you can also act. So bringing big numbers is the wrong approach, in my opinion. You need to show faces and, the fatality of disease. And then I was also embarrassed how cardiovascular diseases is being positioned in public. Poor health, unhealthy lifestyle, drinking, smoking. And then when you see a cancer patient, it's a young lady suffering, and you are full of empathy. So the second, and the third problem we do have is we are always talking, we are healing a patient, or we are putting a stand and helping the patient. Whereas oncologists say, well, we can't heal you, but we help you, okay, or may help you. So our strategy is entirely wrong publicly. And then we ignored the necessity of continuous interaction with regulatory bodies and policymakers, and to join forces across the entire workforce in cardiology. So oncologists always have pharmacy, patients, and academia and clinics jointly. And we were going just as cardiovascular community. And the patients are so important. And we need to focus and join forces with the patients. So, and I think that's just, we need to change that entirely. That's my opinion, and I'm happy it's moving now. It's also moving in many in Spain and in Romania and also in Germany. We are working hard on that, so to change that impression.
And I think you're exactly right, Hugo. It's the individual patient and the individual patient voice, which resonates very, very clearly, because it's not populations that we're dealing with, it's people. And the second is that we don't work in splendid isolation. We work with patient groups, we work with regulators, we work with medtech, we work with all of these people who are all working towards trying to resolve this big challenge that we do have. And I also completely agree that the blame that we put on our cardiovascular patients, it's not lifestyle choices, it's system risk factors that are there for these people that we need to be addressing, as well as what we can do to help people be better. And I'm going to ask you one last question, Hugo, which is a little bit cheeky, but I'm going to ask it anyway. So what's on the menu for you now? Is your priority innovation? Are you looking at AI-enabled cardiology? You've mentioned that. You've talked a bit about molecular therapies. In the past, we've had discussions about digital infrastructure, that kind of thing. So what's on the table for Hugo Katus here at the closing weeks of the end of the first quarter of the 21st century? Looking forward, what comes next?
Not vacation, okay. First, second, I'm still very much involved in these gene therapies stuff. We really try hard, and actually we have quite some successes in developing AAV-associated vectors, which really finds their way to the heart and not to the liver. And so we really hope to have a tool for more cardio-specific therapies on many targets later on. So that's a topic which is important. I'm also still trying to move the field with informatics for life, which is an important, we are building now a new center where informatics for life is part of the smart heart center. And so what will happen is, for years and years, we have bridged digital sciences, AI with cardiology, but then now in the new buildings, they will work together daily. And so the AI man will see a patient and and there will be companies in that. So we really now not only bridge, but we will meld, hopefully, both disciplines, and that I acquired quite a bit of money to realize that. So that was very helpful. And I'm serving the ESC still. I was offered to chair the think tank of the ESC on innovation. And that's very fascinating. Really try to bring in all my experience and have talented people around me and trying to move the field. And so we were in the first part on digital transformation, try to structure a bit in the ESC, and there's a lot of progress, not with us, but anyway, we might help. But the topic right now is I call it systems cardiology. I tell you why. We are going in the future, we have to treat elderly patients, many more. They all have comorbidity, right? And in cardiology, everyone is rushing into subspecialties. And not only in cardiology, it's in gastroenterology, in every discipline. At the end, no one is here again to treat these complex patients, elderly patients, which make up the most of all patients. And we are having in the future more AI and AI assistance. So when a cardiologist uses AI assistance, he will become quite competent in treating complex patients. So our slogan is, and we are moving that and directing that, and we present it to support in the hopefully in the next time, is the slogan is cardiology must remain the heart of internal medicine. All diseases are connected. Cancer impacts the heart. Pulmonary diseases impact the heart. Yeah, inflammation is so we can't ignore that, and we are trying to, and we believe that the term general cardiology is not helpful. Okay, it's interaction of systems, and so we call it systems cardiology, and try to have a new branding and make it more attractive for young cardiologists to remain in the center of medicine, namely as the patient, right? Not as the technology. So that's a bit just
just rebranding the whole of cardiology, just like that. So Hugo, thank you very, very much, and I'm looking forward as we move to the second quarter of the 21st century to see what you're coming up with. So I'm now going to hand back to Filippo.
Okay.
Well, back to your life. I know that one of your friends has been a famous Nobel Prize. Can you tell us?
Yeah. Oh, well, yeah. No, it has to do with what I believe very important relationship of physician with his patients. And I gained so many friends and many important people. And one of my friends a lifetime was Günter Grass, yeah, and he wrote a poem on his heart catheterization. So my name is in world literature, if you want. So so he was very Günter Grass was a very impressive person, well connected in Germany also with politics, but he was actually not only a writer, he was a painter as well. He was an artist. We enjoyed that, his company, and with many others as well. So one message of mine to those who are in clinical care is patients are so important. They are the advocates locally, and of course they may support it surprisingly, and say our heart center in Heidelberg would not have been possible without the support of the patients. We received more than 130 million euros from patients to build that heart center, and then the government added the remaining to 560 million then. So it started. And but I think it's very important to have that partnership and that trust with your patient and dependability, and it's very rewarding, I would say.
Who are the other most important persons from Heidelberg after Kant, Gundergrass, and you?
Yeah. Yeah. So it is. Yeah.
Other important scientists or artists from Heidelberg. Is a famous university, wonderful university.
Yeah. Yeah, you must come to Heidelberg, Sandy.
Okay. Well, very last question. Between one biomarker and the other, what are your hobbies?
Well, actually, I play soccer and I still do sports. Do walking and use my bicycle and work in the garden, looking across Heidelberg from Philosopher's Way. And I changed it a little bit in more passive type of sports. So I'm going to football, soccer game, and every time to our Bundesliga club, which is Hoffenheim. And one of my patients, who has 49% of the shares on that club, he gave me a present to the end of my career, a clinical career, that I have a lifetime 2 tickets for his lounge for every Bundesliga game. And so I'm on Saturday between afternoon, I'm offline and going to soccer. But otherwise, I do enjoy working. I do enjoy science and scientific exchange, and I can't waste time sitting on a beach or so. Or travel around. I haven't learned it. I don't enjoy it actually.
Thank you, Hugo. Let's close on this remark. It has been more than 1 hour, but so enjoyable. Looks like 1 minute, but we spent together more than 1 hour. Beautiful, enjoyable hour. Thank you, Susanna, for your participation, for hosting Hugo. Thank you, Hugo, for this brilliant Socratic conversation touching so many important points. And regarding the future, well, for the first time, we will, after 5 years, we will not have a dialogue in December. As you know, the dialogue is always on the last Thursday of the month. And this year, on the last Thursday of December, will be Christmas. So I thought, well, after 5 years, perhaps we can have this exception, not having the dialogue in December, because it would be in the Christmas day. But we will start again in 2026 on the last Thursday of January at 5 CET, as usual. And you will see, will be another interesting dialogue. Thank you, Hugo. Thank you, Susanna. Thank you, Aloisa. Thank you, all of you, for participating to our dialogue. And if you didn't have time to do it today, the dialogue can be found in X and will be found soon also in YouTube. Thank you very much, and Merry Christmas.